Combustible tobacco claims over 8 million lives annually, making it one of the most thoroughly documented cardiovascular and respiratory hazards in modern medicine. Yet for millennia prior to industrialization, indigenous cultures utilized Nicotiana rustica therapeutically to treat severe respiratory conditions and accelerate wound healing. These traditional healing practices relied entirely on the pure, unadulterated botanical compounds of the plant, avoiding the hundreds of engineered chemical additives found in commercial cigarettes. Archaeological records revealed deliberate delivery mechanisms designed for episodic, ceremonial burning, purposefully avoiding the habitual, continuous chain consumption that characterizes modern addiction. The devastating health crisis we observe today is driven by the toxic mechanics of combustible delivery, which effectively masks the distinct biological actions of the plant's isolated primary alkaloid. Stripped of carcinogenic smoke and tar, nicotine is strictly an isolated alkaloid. In pharmacological terms, it operates as a potent parasympathetic agonist. Recent clinical data highlights an anomaly regarding this molecule. When isolated and administered safely, it demonstrates highly measurable neuroprotective and anti-inflammatory properties. Clinical researchers are now actively testing pharmaceutical-grade isolated nicotine to treat complex autoimmune conditions, inflammatory bowel disease, and severe post-viral syndromes like long COVID. This creates a deep physiological contradiction. A chemical universally recognized as a toxic, addictive stimulant is simultaneously capable of suppressing systemic tissue inflammation. The explanation lies in the molecular architecture of the immune system itself. Nicotine intercepts a specific signaling pathway, reconfiguring how peripheral cells respond to systemic threats. The central nervous system does not operate independently of the peripheral immune system. The two are physically hardwired together. The primary data pathway connecting them is the vagus nerve, which transmits continuous regulatory signals from the brainstem directly down into the visceral organs. This specific biological wiring constitutes the cholinergic anti-inflammatory pathway. It is the body's built-in natural mechanism to keep peripheral immune responses under tight control. To communicate with and modulate the behavior of peripheral immune cells, the vagus nerve releases the endogenous neurotransmitter acetylcholine. Exogenous nicotine perfectly mimics both the shape and function of acetylcholine within this specific biological pathway. By exploiting this pre-existing neural immune bridge, clinical science can control life-threatening systemic inflammation externally. To see exactly how this works, we have to look directly at the microscopic surface of an individual cell. Embedded within the cell membrane is the nicotinic acetylcholine receptor. It is a ligand-gated cation channel constructed from 5 assembled alpha and beta subunits. At the center of this pentameric structure sits a central water-filled ion channel. In its resting state, the pore remains tightly closed, blocking all cellular influx. The extracellular domain of the receptor features precise docking sites waiting to receive specific chemical ligands. The moment nicotine binds with high affinity to these docking sites, it forces a physical conformational change. The 5 subunits twist, causing the central pore to snap open. This initiates the rapid transmembrane flow of positively charged sodium, potassium, and calcium ions down into the cytoplasm. This mechanical opening of the ion channel is the necessary first step to translate an external chemical drug into a profound internal cellular response. Once those ions cross the membrane, the pharmacological action moves inward, mapping to the interior volume of a peripheral macrophage. Inside this immune cell is a transcription factor called NF-kappa B. It functions as the trigger for the body's primary inflammatory alarms. Under normal viral or injury stress, NF-kappa B translocates directly into the cell nucleus to mass-produce pro-inflammatory cytokines, specifically TNF-alpha and interleukin-6. However, the massive ion influx triggered by the nicotine receptor initiates a distinct intracellular signaling cascade. This chemical barrier physically blocks NF-kappa B from entering the nucleus. Deprived of its transcription factor, the nucleus goes dormant, resulting in the absolute cessation of pro-inflammatory cytokine synthesis. This molecular braking mechanism halts the specific protein synthesis cycles that drive runaway inflammation at the source. This reveals a clinical discrepancy. If the molecule functions as a perfect anti-inflammatory mechanism, why is it known globally as a highly addictive stimulant? The answer lies in receptor heterogeneity. Nicotinic receptors are not identical. They are built from entirely different structural blueprints depending on the tissue type. In the central nervous system, the predominant architecture is the heteromeric alpha-4 beta-2 receptor. When nicotine activates this specific brain receptor, it drives dopamine release, cementing the severe reinforcement learning loops that define chemical addiction. The peripheral immune system uses a different structure entirely. Macrophages primarily express the homomeric alpha-7 receptor, built symmetrically from 5 identical alpha subunits. This alpha-7 receptor exclusively mediates the anti-inflammatory cytokine cascade, operating completely separately from the brain's dopamine pathways. Nicotine's dual identity as a neurotoxic hazard and a targeted therapeutic is dictated entirely by which specific receptor subunit architecture it intercepts. Because of this dual interaction, traditional combustible smoking can never be utilized to treat inflammation. Combustible inhalation causes direct, severe systemic tissue injury that immediately eclipses any potential anti-inflammatory benefit of the alpha-7 activation. This graph plots blood concentration over time. Inhalation creates a steep pharmacokinetic spike, flooding the bloodstream instantly and violently hitting the brain's alpha-4 beta-2 receptors to drive addiction. Modern clinical protocols bypass the combustible flaw entirely by utilizing transdermal nicotine application. This chart shows the adjusted absorption curve. By forcing the molecule through the skin, the patch provides a deliberately slow, steady pharmacokinetic profile. This slow release mitigates the extreme dopamine spikes in the central nervous system while maintaining sufficient baseline blood levels to continuously activate the peripheral alpha-7 immune receptors. By altering the route of administration, clinical science can isolate the immunological benefit while significantly dulling the neurological dependency. Ancient indigenous ceremonial use was an early, unrefined exploitation of the alpha-7 regulatory switch. Today, clinical trials targeting autoimmune diseases and long COVID utilize isolated transdermal nicotine to competitively outcompete persistent viral remnants and halt chronic inflammation. But utilizing a non-selective agonist remains imperfect. Even transdermally, nicotine carries off-target cardiovascular effects and lingering addiction risks. The true frontier of modern pharmacology is synthesizing entirely novel, highly specific alpha-7 agonists from scratch. These advanced pharmacological chaperones are engineered to lock into peripheral immune receptors, while physically failing to bind to alpha-R beta-2 addiction receptors in the brain. By reverse engineering the mechanics of a lethal botanical, modern clinical science is synthesizing specific tools to calibrate the human inflammatory response.